This is Chapter 2 of the Instructor's video manual for cloning a fluorescent gene. In this chapter, your students will be performing PCR, polymerase chain reaction, a technique invented in 1983 by a man named Kary Mullis, which eventually earned him the Nobel Prize in Chemistry. PCR is a technique used to take a small number of DNA molecules and amplify that small number of DNA molecules exponentially until you get billions and billions of DNA strands that you can use for further downstream applications. Now that your students have completed Chapter 1 of cloning a fluorescent gene, they should be comfortable using a pipette and transferring liquids into and out of a microcentrifuge tube. This means they're ready to do real biology experiments. Each group of 4 students has already separated out all of the reagents that they're going to need for doing PCR, except for the enzyme, Taq polymerase, which actually does the replication of DNA. Before the students get into class for Chapter 2, you should dilute the concentrated Taq polymerase solution that we've provided you with water, according to the instructions in the teacher's manual that we've provided. As the students are putting together their reactions, each student will individually come up to the instructor's lab bench to add Taq polymerase to complete their PCR reaction. Once they've done this, they'll cap their PCR tubes, initial them, preferably with a black Sharpie, and preferably on the top of the tube and on the side to make sure that it doesn't rub off or get lost in the mass of other tubes. Once they're finished and their tube is labeled, they'll put it in the PCR machine. Once all of the students are ready and have placed their tubes in the PCR machine, you can close the lid, seal it, and begin the PCR run according to the instructions for your specific PCR machine. The PCR machine will take over 2 hours to complete its run, and you can leave it overnight if you'd like. Once you're ready to retrieve the samples from the PCR machine, which should be no later than the following morning, place them in a rack or a bag and store them at 4 degrees Celsius in a refrigerator for up to 5 days. The longer that you store the PCR products, the less active that they're going to become in the ligation reaction. So if you can do PCR on one day and ligation immediately on the following day, that is the optimal situation.